Hello, everyone. Welcome to the session of Animal Kingdom, Theory Part 1. Disclaimer: the images, text, and other material used in our presentations are for educational purposes only. We respect the intellectual property rights of the original creators and owners. If you are the owner and have any concerns or issues, please contact us at sathi@iitk.ac.in and we shall remove the content immediately. Please note that the parts of the content may have been generated using generative AI, which is totally experimental. So, we are going to study today our fourth chapter, Animal Kingdom. And according to the schedule, the chapter 3 that will be conducted after this particular chapter. So, today's learning objective. What is the learning objective of today's lecture? To understand the fundamental criteria used for the animal classification, including the levels of organization, body symmetry, germ layers, body cavity, segmentation, and the presence of notochord. So these are the fundamental characteristics that we must know if we are studying the animal kingdom, because this becomes the basis of classification. Without knowing what is the level of organization of an animal, what is the body symmetry of an animal, germ layer, body cavity, right? All these fundamentals characteristics are very, very important. So this was the first learning objective. Second learning objective: to identify and comprehend the basic characteristic of the primitive animals with a specific focus on the phylum Porifera. So today we are going to study about one of the phylum, that is phylum Porifera, the first phylum, right? And that we call it sponges, and their position in the evolutionary hierarchy of the animal kingdom. So why we keep this phylum Porifera at the very first phylum? Because of the evolutionary hierarchy. We will study detail about this one. Now, basis of classification. This is the 4.1 section of your NCERT book, right? So, in spite of the difference in the structure and the forms of different animals, there are the fundamental features common to the various individuals in relation to the arrangement of the cells, body symmetry, nature of coelom, patterns of digestive, circulatory, and reproductive system, right? So what are the fundamental features? Fundamental features are those features that are present in every organism. So we must know what are the fundamental features. But this fundamental feature in some sort of differences in every organism. Like if we take this circulatory system. So there are two different types of circulatory system: open circulatory system and the closed circulatory system. So in some organism, you will see the circulatory system is open type, and in another, you will see that they are of closed type, right? Similarly, the digestive system. Digestive system may be complete or incomplete, right? So these are the features that we must know before studying animals. And you all know there are so many animals present on our earth, and their study, if you want to study each and every animal, then it will be very, very difficult, right? So we classify organism. Now, what is the basis of classification? The basis of classification is to know about the fundamental features. Not only the fundamental features, there are some specific features also. Okay. So we will study one by one what are the fundamental features and what are the specific features of a particular phylum, right? So today we are going to study about one phylum, that is Porifera. So there I will tell you what is the specific features. Okay. So these features are used as the basis of animal classification. This becomes the base of animal classification. Suppose you get any animal and you want to know about that this animal is like, what is the name and how you know about this animal. So whatever the features you see, you see in that organism, you will note down, you will write it, right? So that will be the fundamental features of this particular animal, right? So here, first of all, we will study about the fundamental features that becomes the basis of animal classification. So, introduction. When you look around, you will observe the different animal with the different structure and forms, right? There are so many types of animals and they're having different structures and forms. Like you see the animals in aquatic environment that are found in the aquatic environment, fishes, they are totally different that the one which you see on the land, right? You are having the mammals, you are having the reptiles, amphibians, right? So there are so many animals, and not only the vertebrates, we must know about the invertebrates also, right? So there are so many animals with the different structures and forms. As over 1 million of species of animals have been described till now, the need for the classification becomes all more important. It is very, very important to know about the classification because it will help you. It will help you to study about such a large number of animals. Look at the number of animals, 1 million, and it will be very difficult to study all these animals, right? If you study one by one. So we follow classification. And what is the basis of classification? The fundamental features, right? The classification also help in assigning a systematic position to the newly described species. So we do not know all the organisms that we see on the earth, right? We don't know about them. So there must be some systematic position to every organism, right? So how to do that? That we are going to study about. In spite of differences in the structure and forms of different animals, there are fundamental features common to the various individual in relation to the arrangement of cells, body symmetry, nature of coelom, patterns of digestive, circulatory, and reproductive system, right? So that are the fundamental features. What are the fundamental features that we are going to study about? Arrangement of cell. What type of arrangement of cell we see in an organism? What is the symmetry of body? What is the nature of coelom? Coelom is the body cavity. We will study about it in a detail. Patterns of digestive, circulatory, and reproductive system. So these are the fundamental features that is present in every organism, but there's some sort of differences. So that is very important to know what some sort of differences are there in the fundamental features in different organisms. So these features are used as the basis of animal classification, and some of them are discussed here, right? So what are the fundamental features that we are going to discuss about? Let us see that. The first one is the level of organization. Level of organization. What is the level of organism? So, though all members of Animalia are multicellular, all of them do not exhibit the same pattern of organization of the cell. All the animals are multicellular because you have studied about the biological classification chapter. Okay? The unicellular organism, eukaryotic one, are kept in the which? Protista. They are kept in the protista, right? But what about the multicellular? So here in the animal kingdom, we are going to study about the multicellular animals, right? So in multicellular organism, what is the level of organization? For example, the first phylum, sponges or porifera. The cells are arranged in loose cell aggregate. What is the meaning of this loose cell aggregate? Let us understand this. They are multicellular, but still, as the cells are loosely arranged, the tissue formation has not been taken place. no tissue organization we see in the case of sponges. It is a multicellular, but still it is showing the cellular level of organization. So cellular level of organization you see in the case of amoeba, you see in the case of paramecium, you see in the case of leishmania, right? But those are unicellular organism, unicellular. So cellular grade of organization. But the important thing is here in the case of sponges that they are multicellular, but still they are having the cellular level of organization. Why so? Because still here tissue formation has not been taken place. Tissue formation has not been taken place, right? So that is why it is showing the cellular level of organization. Some division of labor activities occur among the cells. So they are showing the cellular level of organization. So, but there is a division of labor. Division of labor means some cells are assigned to a work. Like they are having a cell that is called as collar cells or the coinocytes. They are having a very specific function. Okay. There are some cells that are meant for the digestion. There are some cells that are meant for the excretion. But still, tissue has not been formed here. So that is why the poriferans are showing the cellular level of organization. This is very, very important characteristic. We are studying level of organization, the first fundamental features. And in the first, the lowest level of organization is the cellular level. The lowest level of organization is the cellular level. And where we see it in the sponges, porifera, first phylum. Now, in coelenterate is the second phylum. Coelenterate is the second phylum. The arrangement of the cell is more complex. How it become complex? Because here, though they are multicellular, their cells are not loosely arranged. Their cells are compactly arranged. And here, they are forming tissues. So in coelentrates, they are showing the which level of organization? They are showing the tissue level of organization. So here, the cells performing the same function are arranged into the tissue, and hence they are showing the tissue level of organization. So clear it is? Cnidarians or the coelenterate, the next phylum. The first phylum was the porifera, and it is having the lower grade of organization. Lower grade of organization, that is the cellular level. Though they are multicellular, but they are not forming any tissue-like things. In the case of next phylum, coelenterata or cnidarians, their cells are compactly arranged, and many cells unite together to form the tissue, and tissue perform a particular function. So this is the complex one. This is the advanced feature. This is the advanced feature than cellular level of organization. A still higher level of organization. Now, we are also having some higher level of organization, that is the organ level. Now, the tissue, many tissue unite together to form the organ, right? So in the case of the next phylum, Platyhelminthes and other higher phyla, where the tissue are grouped together to form the organ. Many tissue are grouped together to form the organs, and each is specialized for a particular function. It means that every organ is performing a particular function, right? So that is showing the organ level of organization. So we have seen the three level of organization: cellular level of organization, tissue level of organization, and organ level of organization. Now we will see one more level of organization. In animals like annelids, arthropods, mollusks, echinoderms, chordates. I will take one example of each phylum, then you will be able to recall that which organism I am talking about. Annelids. One of the organism is the earthworm. Arthropod, one of the organism is the cockroach. Mollusk, one of the organism is the pearl oyster, the organism which gives you the pearls. Hmm? Echinoderms, starfish, and chordates. Chordates are having the fishes, amphibians, reptiles, birds, and mammals, right? All comes under the chordates. So these animals, what special level of organization these animals are having? They are having system. They are having system. Means digestive system, respiratory system, circulatory system, nervous system, reproductive system. It means they are having system level of organization, right? So if we see from the lowest level, they are having the cellular level of organization, then they are having the tissue level of organization, then they are having organ, and then organ system level of organization. So as you move to the higher organism, higher grade of organism, advanced organism, you will see that they are having some advanced feature in that, because those organisms are more complex. Like we humans are most complex organism, right? And we are having the different system in our body. We are having digestive system, we are having nervous system, we are having circulatory, respiratory, reproductive system, muscular system, skeletal system. So many system we are having. Why we are having so many system? Because our body is more complex, and we require a specific system for a specific physiological function, right? So this is the level of organization. And for classification, to know the level of organization becomes very, very important. So this is one of the fundamental features. So this pattern is called organ system level of organization. Organ system in the different groups of animal exhibit the various patterns of complexities. For example, digestive system in platyhelminthes only having a single opening outside of the body that serve as both mouth and anus. So you see in this particular organism, platyhelminthes. Any example of platyhelminthes can you think about here? Platyhelminthes. Platy means flat. Helminth means worms. You can divide this name in two. Yes, planaria. Very good. Platyhelminthes. Platy means flat, and helminthes, next word is the helminthes means worm. Flat worms. The worms that are flat. So we are having Taenia solium, we are having fasciola. Taenia solium, yes. Tapeworm. The common name is tapeworm. Another example is the planaria that you have given. Very good. So they are having only one opening for the digestive system. Our digestive system completes, alimentary canal complete from mouth and end to the anus. So a complete digestive system must have two opening, mouth and anus. But what about this platyhelminthes? They are having incomplete digestive system. Incomplete digestive system. How can we say that? Because they are not having two opening, mouth and anus. They are having just one opening, that is doing this both the function. That is to take the food also and to withdraw the undigested food material. So they are having incomplete type of digestive system. Very, very important feature. A complete digestive system has two opening, that is the mouth and anus. So the organism that are having the two opening for the digestive system, the through one which you take food, that is the mouth, and the one from which you remove out the undigested food material from the body, that is the anus, right? So that is the complete digestive system. Similarly, the circulatory system. Think about the circulatory system. This is also are of two types. Two types of circulatory systems are there. Open type, in which the blood is pumped out of the heart. Open type. Open type means the blood is pumped out of the heart on the cells and tissue directly. Directly. The blood is coming out from the heart and it is poured all over the cells and tissue. So that is the open type of circulatory system that you see in the case of arthropodans, that you see in the case of insects. Open type of circulatory system. Now we will see the another type of circulatory system. Closed type. Closed type. The blood is flowing in a closed form. Closed form means from the heart, the blood is circulated through every organ with the vessels, with the help of vessels. And they are having the different diameters. So we are having arteries, we are having veins, we are having capillaries. Right? So open and closed type of circulatory system. So we are having this two type of circulatory system, right? So I hope you know what is the meaning of open type. The blood is poured directly all over the cells and tissue. So here the phylum example you want to know. Okay. So one, we are having the arthropodans, right? Arthropodans are having the, and mollusk also. Mollusk also having the open type of circulatory system. In these two phylum, you see that they are having the open type of circulatory system. But when you move to the some of the higher organism, even in the case of annelids, you will see that they are having the blood vessels, closed circulatory system they are having. In our body, the blood is flowing in the closed circulatory system. We are having the definite arteries and veins. Yes, annelida and the chordates are having the closed circulatory system. Though annelida is a lower grade of organism than the arthropoda, but still for the circulatory system, it is advanced than the arthropoda, right? So this is a very, very important to understand the open type and closed type of circulatory system. Example. Now these are the examples of the blood vessels: arteries, veins, and capillaries. Now we will talk about the next fundamental feature. This is also very, very important. Symmetry. So animals can be categorized on the basis of their symmetry. You can divide the number of organisms in the category, or you can categorize them on the basis of symmetry. So sponges are the mostly asymmetrical. Asymmetrical means they do not have any type of symmetry. If you divide the organism from a plane that passes through the central axis, it should divide the organism into two equal halves. But if it is not dividing the organism into two equal halves through any plane that passes through the center of the axis of the organism, then it is called as asymmetrical. So the sponges, that is the poriferans, they are asymmetrical. So this is not a very advanced feature. This is not an advanced feature. The advanced feature we will see later on. Asymmetry. Any plane that passes through the center does not divide them into equal halves. So this is the lower feature. So when any plane passing through the central axis of the body divide the organism into two identical halves, it is called as radial symmetry. So here we can see with the help of this figure. Coelentrates, ctenophores, echinoderms have this kind of body plane. So what type of body plane? You can see, you can divide this organism from this plane into two equal halves. You can divide this organism with this plane into two equal halves. And you can also divide this organism with this plane two equal halves. So there are number of planes that passes through the center of the organism and divide the organism into two equal halves. So such type of symmetry is called as radial symmetry. Radial symmetry. So asymmetrical, that is the first one. Second is the radial symmetry that we see in the case of coelentrates, ctenophores, and echinoderms. This is the name of the phylum in which the radial type of symmetry is common. Okay. So remember the symmetry, radial symmetry, and in which phylum they are found. Now, animals like annelids, arthropods, etc., where the body can be divided into identical left and right. You can divide the organism into two equal halves, passes through the center of the axis. Only one plane. Divide left half and right half equally through one plane only. Then it is called as bilateral symmetry. Bilateral symmetry. Now you can see this figure. This is crab. Crab that belongs to the phylum Arthropoda. So here what you see, this is the plane of the axis that passes through the center axis of the organism, and it is dividing the organism into two equal halves. See the appendages. 1, 2, 3, 4, 5 appendages, and the right side also 1, 2, 3, 4, 5. So equal appendages. Equal appendages. Equal appendages. Yes. Echinoderms. When we will come to the echinoderms, this becomes a very, very important feature of this echinoderms. Right? At the larval stages, they are having the bilateral symmetry, but when it transforms into the adult echinoderms, then it shows the radial type of symmetry. This is a very unique feature. So that is the specific feature. When we will study about a particular phylum, then we will discuss about the specific feature. But here what we are studying the fundamental feature. Fundamental feature. So symmetry is the one of the fundamental feature. We are having three different types of symmetry. Asymmetry means no symmetry is there. The next is the radial symmetry, and the most advanced is the bilateral symmetry. Bilateral symmetry. Now, diploblastic and triploblastic organization. This type of organization, what is actually this diploblastic and triploblastic? Actually, when there is a formation of embryo, there are number of cells that arrange themselves into a layers. Okay? Embryo formation, I'm talking about like zygote forms there after the fertilization, and zygote divide and redivide and forms the number of cells. Now, what happens to this number of cells? The number of cells arrange themselves into layers. They arrange themselves into layers. So, the outermost layer, the innermost layer, and the layers in between the outermost and innermost. On the basis of this arrangement, we are having the two types of organization, diploblastic and triploblastic. Let us see that. So the animals in which the cells are arranged into the two embryonic layer. At the time of embryo formation, the cells are arranged in two layers. The outer layer or the external layer, that is called as external ectoderm, and the inner layer that is called as internal endoderm. Only two layers are there. So such type of organization is called as, and such type of animals are called as diploblastic animals. Okay, so an organization is called as diploblastic organization. The animal in which such type of arrangement we see, we call them as a diploblastic animals. And one of the example is the coelentrates, like hydra. Okay, an undifferentiated layer, mesoglia, is present in between the ectoderm and the endoderm. There is one layer that is called as mesoglia. Mesoglia is present in between the external ectoderm and internal endoderm, but it is not a cellular layer. So here you can see, this is the mesoglia layer. This is a jelly-like substance that is present in between the ectoderm and endoderm. That is called as mesoglea. So this is a diploblastic organization. Now, those animals in which the developing embryo has a third germinal layer. Now, here they are having the third layer, the mesoderm, in between the ectoderm and endoderm. And that is why it is called as triploblastic. Triplo means triple layer. Blastic means layers of cells. Means 3 layers of cells are there. Now, they are quite advanced. The organisms in which the embryo them arrange themselves into 3 germ layers, that is the ectoderm, outer one, endoderm, inner one, and in between the ecto and endo there is a middle layer that is called as mesoderm. Now here the derm is there. In the case of diploblastic, we are having mesoglea. Mesoglea, that is not the cellular layer. But yes, this mesoderm is a cellular layer. So let us see the figure. That gives you the clear-cut understanding. Here you can see. This is the ectoderm, outer ectoderm. And this particular layer, this layer. This to this. This layer is the endoderm layer. What about this red and this peach color? That is the mesoderm layer. Okay? The third layer in between the ectoderm and endoderm, that is the mesoderm layer. This is the cellular layer. And this is found in between. So from which phylum you find them? Platyhelminthes. This triploblastic layer we see from the platyhelminthes up to the chordates, the most advanced one, right? So this is the advanced feature. Triploblastic. This is the advanced feature. Diploblastic is not an advanced feature. Now, very, very important thing, coelom. Coelom means body cavity. So the presence or the absence of cavity between the body wall and the gut wall. What is the gut wall? We are having the alimentary canal now. So this alimentary canal is called as the gut, right? The gut. And the wall of the gut and the wall of the body, in between these two, body wall and gut wall, there is a space present or no space present. If a space is present, then you say that they are having the body cavity. And if no space is present, then no body cavity, right? So the organism which possesses this space in between the body wall and gut wall, they are called as coelomate animal. And the cavity is called as coelom or the body cavity. And the organism in which there is no cavity present between the body wall and the gut wall, that is called as acoelomate. So the gut wall, you want to understand the gut wall. Gut is your like stomach actually, right? And this, if you think about this alimentary canal, then it is start from the mouth and end to the anus. And what this canal like system you see there? So you see the mouth, you see the esophagus, you see the stomach, you see the small intestine, large intestine, rectum, and then comes the anus. Is this cavity is having the same diameter? No, it doesn't, right? So according to the digestive function, the diameter of this alimentary canal varies, right? So this, we are having a cavity-like structure which is called as gut, right? Your gut. And that the wall of this gut is called as gut wall. Like we are having the body wall. The body wall. Okay? So the body cavity which is lined by the mesoderm is called coelom. This is very, very important to understand. So here you can see, this is, if I say this is the body wall. Body wall. And here we are having this like gut. This is the gut. And this is the wall of the gut. This is the wall of the gut. So GW means gut wall. Now there is a cavity in between this. And if this cavity is lined by the mesoderm, The body wall inner lining, this inner lining is of mesoderm, and here the gut wall lining is also made up of mesoderm. Then it is called as coelom or the true body cavity. Okay. Animal possessing coelom are called as coelomates. The animal which are having true body cavity, they are called as coelomates. So annelids, mollusk, arthropods, echinoderms, hemichordates and chordates, all are coelomate. All are coelomate. So here you can see. Here you can see. This is the body wall. This is the body wall. And this is the gut. This is the alimentary canal. You can see this is the gut. What is the outer lining of this gut? The red color you see. The red color, that is the mesoderm. And here the body wall. What is this inner lining? This is again mesoderm. So the cavity, this is the cavity, white color. This is the cavity. This cavity is true cavity. That is coelomate. Because from both the side you see, the body wall is also having the mesoderm layer, and the gut wall, outer layer is again, they are having the mesoderm layer, right? So because of this mesoderm presence is very, very important. Okay. If they are not having the mesoderm, then it is not a true body cavity. This is also very important. Okay. So here, these are the coelomates organism in which they are having this mesoderm layer, coelom body cavity. Now in some animals, the body cavity is not lined by the mesoderm. Instead, the mesoderm is present as a scattered pouches in between the ectoderm and endoderm. Let us see that. And such body cavity is called as pseudo-coelom. Pseudo means false. This is not a true coelom. This is a false coelom, false body cavity, right? And the animal possessing them are called as pseudo-coelomates. They are called as pseudo-coelomates. For example, Aschelminthes. Aschelminthes are roundworms. Ascaris is the example. Roundworms. So this is, you see, the pseudo-coelom, the body cavity. This is your gut. This is the gut. Is the outer lining is here? The outer lining of the gut is having the mesoderm layer? No, they are not having. They are not having. Okay. But here you can see, this is the mesoderm layer. This is the mesoderm layer. Again, this side, the mesoderm is not present. So these body cavity are pseudo-coelom type. Body cavity is there, but they are present in the small pouches. And the cavity is not lined by the mesoderm. So such cavities are called as pseudo-coelomate. Such cavities are called as pseudo-coelomate. Okay. And the very good example is the Aschelminthes roundworms. Now the animal in which the body cavity is totally absent, no body cavity, acoelomates. That is the Platyhelminthes. The Platyhelminthes are not having their digestive system. They absorb the digested food material from their body surface. So that is why they do not have this digestive system. So that is why they are not having any body cavity as well, right? Acoelomate, Platyhelminthes. So we are having the 3 worm phylum. Very, very important thing. Listen carefully. 3 worm phylum. The first one is the Platyhelminthes. No coelom is found. Next phylum is the Aschelminthes, roundworms. Pseudo-coelom is found. And the third worm, Annelida, they are having true coelom. They are having the true coelom. And from the Annelida to the next level, whatever the phylum after that it is coming, they are all having the true coelom, true body cavity. Clear, I hope? This is very, very important fundamental feature, the presence of coelom, right? So here you can see the diagram of acoelomate. They are having ectoderm, they are having this endoderm, they are having this mesoderm. but they're not having any cavity. They're not having any cavity. So this is the diagram of the acoelom. Next is the segmentation. Segmentation. In some animals, the body is externally and internally divided into the segment. You see, there is a segmentation from the outer side. There is a demarcation of the segmentation. Like you see in the case of arthropods, you see in the case of Annelida, earthworm, very good example, in which you see the segmentation. The body is divided into small segments. That you see from the outer side. But when you cut the organism and you see from inside also, they are again having from inside also. They are having the segmentation demarcation from inside also. From outside, from inside. Both the side, you can see there is a segmentation line. And in each segment, what you see? Repetition of at least some organ. There is a repetition of organs in the segmentation. This is a very, very important feature. And such type of segmentation, such type of pattern is called as metameric segmentation. This is called as metameric segmentation, right? And the phenomena is known as metamerism. This is called as metamerism. Next fundamental feature is the notochord. Notochord. So notochord is a mesodermally divided rod-like structure formed on the dorsal side during the embryonic development in some animals. So this is the mesoderm origin, rod-like structure, and which side it is present? In the dorsal side. Either it is present in the embryonic development stage or in the later advanced stage also. So the animal with notochord are called as chordates. Animals in which the notochord is found or present, they are called as chordates. And the animals in which do not form this structure, non-chordates. Non-chordate. So from the porifera to echinoderms, no notochord is present. Okay, no notochord is porifera, coelenterata, platyhelminthes, esculenthes, Annelida, arthropoda, mollusca, echinodermata. They do not have any notochord either in the embryonic stage or in the adulthood stage. No notochord is present. So that is why they are called as non-chordates. What is the difference between the nerve cord and notochord? When we will study about the chordates, I will come to that point. Okay? Notochord and nerve cord. Right? We will discuss at that time. Now, the broad classification of Animalia based on the common fundamental features are mentioned in the preceding section given in the figure. Notochord is a rod-like structure that is meant to the nervous system. Notochord is meant to the nervous system, and this is either present in the embryonic stage or in the adulthood stage. But from the phylum Porifera to the echinoderms, you will not found any notochord presence. No notochord is present, and that is why they are called as non-chordates. The organism in which this notochord is present, that is called as chordates. Just remember this. When we will study chordate, then we will discuss this in great detail to understand notochord, vertebral column, nerve cord. All we will understand there. But till here, you just remember notochord presence, that is called as chordates. Notochord absent or not found, that is non-chordate. Okay? Okay, now coming to this particular figure, this is very, very important. So important characteristic feature of the different phylum are described here. So here you can see. This figure makes you understand why this fundamental features are important. So here you can see what we are studying, Kingdom Animalia. All are multicellular organism. Yes, we are studying multicellular organism. We are not studying unicellular. Level of organization. There are two levels of organization. Cellular level, porifera. And we are having tissue level of organization, we are having organ system level of organization, and we are having organ system level, right? So this system, all this advanced, we are keeping it together, but this cellular level we are keeping aside. Coming to the symmetry, mostly is asymmetrical, and we see three different types of symmetry: asymmetrical, radial symmetrical, and bilateral symmetrical, right? Okay, coming to the body cavity, porifera, no coelom is found. Radial also, no coelom is found. And bilateral, bilateral also, we are having one category without any coelomate, we are having one category with pseudocoelomate, and we are having category coelomates, true body cavity, right? Now see the phylum, porifera. So the organism which is having cellular level of organization, asymmetrical, acoelomate, they are called as porifera. The organism which are having the tissue level of organization, radial symmetry, acoelomate, they are two types. Two phylums are there, Coelenterata and Ctenophora. Now bilateral, you see the three different categories on the coelomate: acoelomate, platyhelminthes; pseudocoelomate, escalminthes. Coelomate and bilateral symmetry, we are having Annelida, Arthropoda, Mollusca, Echinodermata, Hemichordata, and Chordata, right? Now we will study porifera. First phylum, porifera. So members of this phylum are commonly known as sponges. Okay, phylum are commonly known as sponges. They are generally marine and mostly asymmetrical. Generally marine means not 100% organism are found in the marine environment. There are some organisms that are reported in the freshwater environment. They are found, some organisms are found in the freshwater environment. And asymmetrical. Now here you understand why we have studied the fundamental feature. If we have started from the porifera, then you will not be able to understand what is asymmetrical, what sir is talking about. Asymmetrical means the body is not having any symmetry, right? So here you can see, if you divide the organism from here, it is not showing any symmetry. If it divide the organism from here, it is not showing any symmetry, right? So asymmetrical animals. These are primitive multicellular animals and have cellular level of organization. All the organisms are multicellular, but they are primitive. Primitive means very old organisms, right? They are multicellular, but what type of organization we see here? Cellular level of organization. Cellular level of organization. They are not free swimming. They are attached to the ground, means they are sessile. The particular term here you use for this attached organism, that is called as sessile. Sponges have a water transport or canal system. They are having water transport or canal system. Very, very important feature of this particular organism. Now, for this water transport and canal system, you see minute pores on its body surface, that is called as ostia. Okay, this ostia leads to a central cavity which is called as spongocele. So the organism is like this. I'm just drawing a rough figure to understand this. Okay, and inside this is a cavity like thing. So they are having minute pores here. From these pores, the water get enter here and it lead to this central cavity. This is the central cavity or spongocele. This is the spongocele cavity. So water enter from this ostia, and this ostia is present all over its surface. It is present all over its surface. So water enter from this ostia, makes its way inside to this spongocele, right? And here at the mouth, this is the opening that is called as osculum. right? This is the osculum, and this is the ostia. So this makes the central cavity. This is the central cavity, and this makes the canal system. From ostia, the water is entering in the central cavity or the spongocoel and coming out from this osculum. Okay. So this pathway of water transport is helpful in food gathering. Okay. This water transport is helping in the food gathering, capturing of the food, respiratory exchange means gaseous exchange, and also the removal of waste from the body. So this canal system or water system is very, very important for, because it is helping in the capturing of food, respiratory exchange, gaseous exchange is there, and also the removal of the waste material from the body. Poinocytes. Now, I told you that there are some specific feature of the phylum. So this is the specific feature of this phylum. Poinocytes. Such cells are only found in the porifera. Poinocytes are not found in any other phylum, right? So this is the specific feature of the phylum Porifera, right? Coinocytes or collar cells are the lined in the spongocoel cavity. It is lined in the spongocoel cavity. So what is the structure of this? It is something like this. This is the collar-like thing, and they are having the flagella, right? So this is the collar. This is the collar. Collar. Okay, this is the collar cells. And they are lined, they are present in the spongocoel cavity. They are present in the lining of the spongocoel cavity. So they can capture the food material, they can take out the waste material from that, right? Digestion is intracellular. The body is supported by the skeleton made up of the spicules and spongin fibers. They are having some sort of skeletal system because of the presence of this spicules and spongin fibers, right? That makes the body of the sponges. Sexes are not separate. Hermaphrodite means the organism is having both the organ. Both the organ means male reproductive organ as well as the female reproductive organ. So such organism in which both the male and female reproductive organ are found, they are called as hermaphrodite. Hermaphrodite. They gives rise eggs and sperms and produce the same individuals. Sponges reproduce asexually. Sometimes what happens, the sponges can break down and each part will behave as a new sponge, right? So there is a process of fragmentation through which the asexual mode of reproduction is we see. Okay, sexually they forms the gamete also. Fertilization is internal and the development is indirect, having the larval stage which is morphologically distinct from the adult. Examples, let us see the examples. Cycon, this is the scientific name. Scypha is the common name. Spongilla, here you can see the freshwater sponge is there. Euspongia, that is the bath sponge. So these are some of the examples you must remember. So these are the examples with the diagram. Summary. The basis of the animal classification, what we have studied about the basis of classification, that is the symmetry, okay, presence or absence of the body cavity, that is the coelom, segmentation, notochord, and number of the germ layers. Phylum Porifera, commonly known as sponges, represent the most primitive multicellular animal characterized by the porous body canal system. And they are having the coinocyte, that is the specific feature of this. They are having the unique water current system for the feeding and respiration. These sessile, they are sessile organism. They are attached to the substratum, right? They lack true tissue or organ, possesses a skeleton made up of the spicules or spongin fibers, demonstrate the remarkable regenerative abilities while the primarily reproducing both asexually and sexually. So this is all about today's session. Just give me a minute. Okay. So we will do the DPP as well. Don't worry. We will have DPP. Okay. So this is the first question. Animal which possesses collar cells and an osculum are characteristic feature belong to the phylum. So this is the first question. Animal which possesses collar cells and an osculum are characteristic feature belongs to the phylum A. Nidaria, B. Echinodermata, C. Ctenophora, and D. Porifera. Right? So, which we have just studied that the collar cells are the characteristic feature of the Porifera, right? So, we'll opt for the Porifera, save, and move to our next question. So, the next question is here. Which level of organization is exhibited by the coelentrates? Level of organization is exhibited by the coelentrates: cellular level, tissue level, organ level, and organ system level. In Porifera, we have studied about that they are not having any tissue level of organization or the organ level. So they are having the cellular level. But the very next phylum, coelentrates, they exhibit the tissue level of organization. They exhibit the tissue level of organization. So we will opt for the option B, save it, and move to the next question. So the next question is here. Which symmetry is observed in animals where the body can be divided into identical left and right halves in only one plane? Through one plane, the organism can be divided into left and right. So options are asymmetrical, B. radial symmetry, C. bilateral symmetry, and D. spherical symmetry. So everyone says bilateral symmetry. Yes, very good. The organism can be divided into two equal halves, left half and right half, equally through one plane only. Okay, so that is the bilateral symmetry. Next question. Triploblastic animals are characterized by the presence of which germinal layer? Triploblastic. 3 germ layers are there. Echinoderms only, ectoderm and endoderm, C. ectoderm, endoderm, and mesoderm, and D. mesoderm only. So in which we see the triploblastic, the organism which are having the 3 germ layer as a ectoderm, endoderm, and mesoderm, right? So we will opt for this, save it, and move to our next question. Pseudocoelomates are the organism that have a true coelom lined by the mesoderm, A. No body cavity, that is the B. C. A body cavity not lined by the mesoderm, and option D. A coelom divided into segments. So pseudocoelomates, not lined by the mesoderm. Very good. C option, a body cavity that is not lined by the mesoderm. They are having the body cavity, but it is not lined by the mesoderm layer. So that is the pseudocoelomate, false body cavity, right? So we will opt for this and save it and then submit it and see the results. So yes, all the answers are correct. Okay. So this is all about today's session. Okay. Thank you very much for attending today's session. We will meet again tomorrow. Okay. So goodbye and take care.